Hi everyone, my name is Xiaosai. I'm from Genentech. So today I'm going to introduce this method called Epiregulon, and that is for inference of transcription factor activity at the single cell level to help us understand lineage transitions and drug response. So as a pharmaceutical company, we are very much interested in drugging transcription factors and transcriptional co-regulators. This is a list of targets that are either approved or currently being evaluated in the clinic. So we are very much interested to utilize gene regulatory network inference methods to nominate new targets and also to evaluate our therapeutic agents. However, there are several limitations with the current GRN inference methods. The first of which is that current methods do not really account for situations in which the TF expression is decoupled from the activity. So you may be more familiar with example involving lineage specification in which the transcription factor is turned on when the transcription factor itself is transcribed. However, with drug perturbations, even though the transcription factor is transcribed, you have small molecules that can interfere with the protein function. In this case, it's depicted as evicting the protein from binding to the chromatin. So therefore, GRN methods that rely on TF expression may mislead the TF activity inference. And then the second limitation is that there's this class of transcriptional co-regulators that do not bind to the chromatin directly, but instead interact with different transcription factors in a highly context manner. So therefore, methods that rely on motif annotations may not be able to account for these class of targets. And then lastly, we all know that there's a paucity of ground truth data that can be used to benchmark our GRN inference methods. So we introduced Epiregulon. It is a method to predict TF activity from single-cell ATAC-seq and RNA-seq data. First of all, we construct the GRN using, first of all, by linking the target to the peaks based on the correlation between the target gene expression and the accessibility. And next, importantly, we use ChIP-seq data to determine the TF occupancy. And this allows us to extend the inference to the transcriptional co-regulators lacking motif. And then we perform an optional pruning step in which we retain only the regulatory relationships if the transcription factor expression, the chromatin accessibility, and target gene expression tend to occur in the same cells. So this constructs the initial unweighted GRN. And then next, we estimate the weights of the regulatory relationships using 3 different methods, but the default is this Wilcoxon method. So the weights equals to the test statistics of this Wilcoxon test, which compares the ranks of the target gene expression between 2 groups. The first group is the active group of cells in which we require concomitant target transcription factor expression and chromatin accessibility. And then compare that with the inactive group in which either of the criteria was not met. And lastly, we hope to provide some functional annotation to the transcription factor function by performing gene set enrichment of the target genes, as well as to understand what are the potential interaction partners using the overlap in the target genes, and also provide differential network analyses using edge subtracted networks. So Epiregulon comes in 3 different R packages. The core Epiregulon package accepts single-cell experiments. And then if you are interested in using Archer projects, since you process the data with Archer, we also have a second package allows integration with the Archer project output. And then lastly, we have the Epiregulon Extra package that allows you to visualize the data and perform differential network analyses. So now we proceeded to generate our own ground truth data, and we use this technique called reprogram-seq, which allow us to introduce defined transcription factors into cells and then track the identity of the transcription factor that each cell has been introduced to. So in this case, we introduce 2 factors, NKX 2.1 and GATA6, into LINCAP, a prostate adenocarcinoma cell line. So if you just look at the gene expression you can barely see NKX 2.1 expression. You can vaguely see the expression of GATA6 in cluster 1. However, if you look at the activity, which is the sum of all the target genes we infer, you can really see NKX 2.1 activity lighting up in cluster 3, and then GATA6 activity lighting up in cluster 1. And this does correspond with the hashtags, which actually marks the ground truth because we know where the cells actually came from. So Epiregulon helps to enhance the detection limit of transcription factor activity when the gene expression of the transcription factor itself is very low. Because of the use of ChIP-seq data, Epiregulon is able to recover the most comprehensive collection of regulators. So compared to other GRN inference method, Epiregulon is the only method that's able to retrieve and predict NKX 2.1 activity. For GATA6, most of the other GRN methods could also calculate this ROC curve, which is defined by the ability of the TF activity to cluster the cells according to the correct barcodes. And in this case, Epiregulon is ranked number 2. We also asked the question, what is a function of GATA6, the second transcription factor we introduced? GATA6 does not have well-known functions in prostate cancer. So we turn to understand its function by looking through its interaction partners. We immediately saw that FOLSL2, which is a paralog of FOLSL1, YAP1, and TEAD1, are amongst the top interaction partners. And interestingly, these factors have been previously associated with reprogramming prostate cancer cells towards a stem cell-like phenotype. And indeed, YAP1 and GATA6 have a huge overlap in the target genes. So we asked, is it possible that GATA6, by virtue of its association with these reprogramming factors, is also able to reprogram the prostate cancer cells? And indeed, we look at these regions that are uniquely accessible in a stem cell-like subtype defined by a separate publication. We saw that GATA6 overexpression can induce the chromatin accessibility at these stem cell-like regions. So we show that Epiregulon is able to uncover a new function of GATA6 in its ability to reprogram prostate adenocarcinoma towards a stem cell-like subtype. So thus far, the reprogram-seq example illustrates how we can use Epiregulon to estimate TF activity when the transcription factor activity is coupled to the gene expression. But a more challenging task, as I have alluded to earlier, is situations in which the TF activity is decoupled from expression. So we generated a second data set in which we treat 4 different prostate cancer cell lines with 3 different drugs against androgen receptor, either directly inhibiting it or indirectly inhibit it. So the first drug is called enzalutamide. It is an AR antagonist. It blocks AR at its ligand binding domain. And then the second drug is an AR degrader, so it degrades the protein itself. And the third one is a degrader of SMARCA4, which is an ATPase subunit of the Swine-Steve chromatin remodeler, and is important for recruiting androgen receptor through the chromatin. And then we treat 4 different prostate cancer cell lines. 2 of them are AR dependent and 2 are AR independent. So using the 2 AR dependent cell lines, VCAP and LINCAP, we know that these cells should respond to the AR inhibitors by decreasing the AR activity. However, if you just look at the AR gene expression, you can see that you don't see much changes. In fact, in VCAP cells, enzalutamide tends to increase the AR gene expression. However, if you look at the AR activity, you can see that Epiregulon correctly predicted a decreased AR activity across all 3 drug modalities in both of the cell lines. Then we took this data set to benchmark all the pretty high-profile GRN inference methods, and you can see there's a range of performance. And Epiregulon is the method that's able to consistently predict a decreased AR activity across both cell lines. In fact, many of the other methods that rely only on expression even predicted the reverse trend. And then lastly, I mentioned that Epiregulon is able to extend the inference to transcriptional co-regulators lacking motifs. And then the third drug is actually a degrader of Swine-Smith chromatin remodeler, which is an example of the chromatin regulator lacking motif. So you can see here that we are able to predict a decreased decreased activity of SMARCA4 across all 4 cell lines, and none of the other GRM methods is able to make predictions for this particular chromatin remodeler. But the more interesting question is, what are the interaction partners of SMARCA4? Since this is a chromatin remodeler, so it is highly context dependent. So, so we make use of the Jacquard similarity of the overlap in the target genes, and we found that in the AR-dependent cell line LNCAP, the androgen receptor is the top interaction partner, and androgen response is the top pathway. So SMARCA4 in this case interact with AR. In another cell line that is AR independent with neuroendocrine features, H660, you can see that the second highest interaction partner is TCF12, which is a factor important for neuronal development. And then EZH2 is also one of the top 20 interaction partner, and EZH2 has been previously shown to reprogram prostate cancer cells towards neuroendocrine differentiation. And the other partners are subunits of the transcriptional machinery known to interact with SMARCA4. So in summary, hopefully I've shown you that transcription factor and co-regulators are important class of oncology targets. And then Epiregulon is able to cover new biology by predicting drug response, especially transcriptional co-regulators lacking motifs. So none of this work would have been possible without my collaborators. I only highlight some of them here. Tomek, Aaron, Russell, Bob, and Ryan. So the package is now released on GitHub together with its preprint. So please check it out and feel free to reach out to me. One more thing is we just have a postdoc position open. So if you're interested, please feel free to reach out. And yeah, okay. And thank you. Thank you. Yeah.
Any questions? Yeah. So I don't know where I'm supposed to. Anyway, the reprogram-seq is a very interesting method. Is it like is there a paper associated with it? Is it genetic specific
Can you repeat the question?
thing?
Oh, whether reprogram-seq is published? Yes, it is published. So so if you check out my preprint, there's a reference. So it's published in 2019, I believe, in Molecular Cell. Yeah.
Okay.
Yeah.
I have another comment. So you said that, like, TF activity-based methods are actually, people are actually ignoring it. So there's a whole class of methods where people are actually trying to estimate activity. So if you look at transcription factor activity, inference of those, there's in current GRN inference methods actually explicitly trying to incorporate activity, estimate activity to infer GRN. So just, maybe take a look at that. I'm happy to point you to some references as to how actually people are trying to estimate the decoupling of the mRNA to the activity level.
Yeah, that would be great. Yeah, thank you.
I had a question on one of your RSG curves. Cell oracle was actually, it seemed like if you just did the reverse predictions, it might have had the highest AUC.
Yes.
So is that a good method, just to reverse cell oracle's predictions?
So, well, the question is, Cell Oracle actually see a reverse RC performance. Is it okay just to reverse the area under the curve, or the 1 minus that? No, because, first of all, this is showing just the enzalutamide, the response to enzalutamide. And if you recall, we have 2 other AR drugs. And enzalutamide is the one that increased AR expression after treatment as a way of compensate, I think, trying to compensate. But the other drugs do not actually cause an increase in AR gene expression. So for this particular drug, you can, but not this. It's not the same. It's not consistent for all the other AR inhibitors.
Okay, thank you.